Hi, everybody. It's Dr. Lonville. This is online podcast number 4. We're going to continue our walk through a bunch of different signaling pathways. This is one that is not talked about much in your book, but it is super important. You run into it a bunch in primary literature papers, and so you should know about it, and that is nitric oxide signaling. So it's a second messenger, but instead of being a soluble ion or a soluble small molecule, it's a soluble gas. It can act as a neurotransmitter. It's important for formation of short-term memory. We'll talk about in short-term memory and long-term memory. We'll talk about long-term potentiation in this podcast. It's a vasodilator, and as a vasodilator, is important in penile erection. We'll talk about that in this podcast. And by manipulating nitric oxide signaling, you can manipulate behaviors such as coordination. So if you give mice a drug to inhibit nitric oxide synthesis, and then you make them walk on a tightrope, then it's not part of a bizarre little circus, but it's a test for coordination, where they have some kind of food reward at the end of the tightrope, and the mice are walking across that tightrope. If you inhibit nitric oxide synthesis, they can stay on longer before falling off. In male mice that are caged together, wild-type mice will usually have some fights, and then they'll establish a dominance hierarchy, and they'll say, okay, that guy over there is the chief mouse. But if you knock out nitric oxide synthesis, they just keep on fighting until they die. It can regulate intracellular ion concentration, especially calcium, and it can affect the cytotoxic activity of macrophages, and we'll talk about that toward the end of this podcast. So, how do you make nitric oxide gas? You make it with an enzyme called nitric oxide synthase, or NOS. Uses arginine as a substrate, produces citrulline and nitric oxide. This chemical reaction requires NADPH, it requires calcium, calmodulin, which is a calcium-binding protein, and oxygen, and that's important for some of the signaling we'll talk about. There are many different kinds of nitric oxide synthase. Here's a 3D model of nitric oxide synthase. There's a form made in neural tissue called neuronal NOS. There's a form that is expressed inside of blood vessels called endothelial NOS. And then there's a form that is ubiquitously expressed that is made as a result of signaling that's called inducible NOS. eNOS and nNOS, endothelial and neuronal NOS, also sometimes called cNOS, because they're constitutively expressed. They're always has some kind of background expression. And cNOS as opposed to iNOS, which is made in response to a stimulus. iNOS is cytoplasmic, and eNOS is associated with insoluble cell structures. So if you're going to do some kind of centrifugation experiment, eNOS would be in the pellet of the cell prep, and iNOS would be in the supernatant of the cell prep. nNOS would be found in both. NOS contains a heme group. It's important to the function of nitric oxide synthase. Here's the heme group in the binding pocket of nitric oxide synthase. So in neurotransmission, nitric oxide synthase signaling is important, and it is related to why repetition allows you to remember things. This is called long-term potentiation. This is pretty complicated. I'm going to show you two different diagrams that they both have their virtues in terms of how well they explain something, and then I'll give you a link to a video that I think does an okay job also, but no one of these does mentions all the things I want you to know. So you have a chemical synapse, presynaptic cell, postsynaptic cell. The presynaptic cell is releasing a neurotransmitter. In this case, it's glutamate as the neurotransmitter. That glutamate binds to the postsynaptic cell in two different kinds of receptors. NMDA glutamate receptors and non-NMDA glutamate receptors. These, you'll see in the next slide, are called AMPA receptors. So initially, the NMDA receptors don't respond to glutamate in that they don't open to glutamate. They are blocked by an ion in the channel, a magnesium ion in the channel that prevents conductance. But the non-NMDA receptors do respond to glutamate, and they open up and conduct sodium. As they conduct sodium, they induce depolarization in the postsynaptic cell and action potential in the postsynaptic cell. If you get continued depolarization in the postsynaptic cell, it reaches a certain threshold, and that threshold will eventually knock off the magnesium ion. Now that the magnesium ion is gone, both the NMDA receptor and the non-NMDA receptor now both respond to glutamate, and that they open up. And in the NMDA receptor, when it opens up, it'll conduct calcium and sodium. Okay? So you have postsynaptic cells, action potentials being generated, and the calcium that comes in to the postsynaptic cell is a required signal for activation of nitric oxide synthase. C requires calcium. Okay, so nitric oxide synthase is activated, and nitric oxide is made. And because it's a gas, it'll diffuse across membranes, and it actually diffuses back to the presynaptic cell. And the nitric oxide that travels back to the presynaptic cell makes it more likely for that presynaptic cell to release glutamate. So, the other part I'll explain in another section. But here you have an idea that a little bit of stimulation of this pathway, and you get postsynaptic cells sending off an action potential. But if you have a lot of stimulation of this pathway, the postsynaptic cell changes such that this whole pathway is more likely to fire off an action potential. Because the nitric oxide is being released after you've had a lot of action potentials go through this system, that makes this much more likely to happen. So as you activate a synaptic pathway, activate synaptic transmission from one neuron to another, say repeating something, saying trying to remember this pathway, then when you are trying to recall that, it is now more likely to fire off that pathway, and that's called long-term potentiation. So this effect can last hours to days, in that once nitric oxide is released and you want to activate that pathway again, it's easier than when it was a novel pathway, brand new pathway, and that easier effect can last hours to days. Here's another diagram from a different source called Study Blue. Here's the presynaptic cell, here's the postsynaptic cell. In this case, they're labeling NMDA, and the non-NMDA receptor is an AMPA receptor. Okay, so just to review, glutamate is released. When glutamate is first released on the postsynaptic cell, only the AMPA receptors react to the glutamate, and the NMDA receptors don't because they're blocked by magnesium. But if you have a lot of action potentials, excitatory postsynaptic pulse, EPSP, okay? A lot of action potentials are generated in the postsynaptic cell. That'll knock off the magnesium. Now NMDA can get in on the game, and calcium comes in, and also sodium through this same channel. Not showing that in this diagram. That's why I'm using several diagrams. That calcium that comes in activates the synthesis of nitric oxide. Nitric oxide diffuses to the presynaptic cell, makes it even more likely for glutamate to be released. In addition to all that stuff, there are AMPA receptors in vesicles that travel through the Golgi apparatus, ready to go, but they haven't fused with the cell membrane. And this process of activating the NMDA receptor causes these AMPA receptors to be fusing with the cell membrane. So now the postsynaptic cell is even more sensitive to glutamate than it was before long-term potentiation. So in addition to the nitric oxide making it more likely for glutamate being released from the presynaptic cell, it also makes more AMPA receptors available on the cell surface in the postsynaptic cell, so the postsynaptic cell is more sensitive to glutamate. And again, making it more likely that this synaptic transmission will happen. If in an LTP neuron, you get a bigger response than in a normal synapse, okay? And if you use an inhibitor to NMDA, you inhibit learning. And this has been shown in all kinds of different models. There's a 2-minute video on YouTube. Here's the link right here. I encourage you to take a look at that. It's animated. It's a pretty good explanation. Doesn't go over all this stuff, so it doesn't have all that I want you to know in there. But if this is troubling you, I encourage you to look at it. Okay. Because of the study of long-term potentiation and many other neurological pathways, we're actually getting a lot closer to understanding how memories form and how to access memories. There is a technique called optogenetics where you can insert in cell neurons of the brain of rodents a ion channel that is sensitive to light. So it conducts when it's stimulated by light. And therefore you can fire off a particular synaptic pathway by giving these cells a pulse of light. And in scientists who are trying to understand the cell physiology of memory, they've used a rat model where these rats are conditioned to react in fear when they hear a certain sound. So they condition them that they play a tone, and then right after they hear that tone, they receive a shock. And eventually what happens is that the rats become entrained to that, and as soon as they hear the tone, they freeze because they say, okay, something bad is going to happen. And these scientists understand where in the brain of these mice, what cells in the brain of these rats, what cells has this been encoded in their long-term potentiation. And then they express ion channels to activate those cells, and they can activate those cells by a pulse of light. And this is a little fiber optic source of light going right into the brain of these cells, such that without a tone, they can just activate that cell with a pulse of light, causing ion conduction and nerve impulse, action potential, and they can get the mice to freeze. So they can get the mice to recall that memory, even though they're not actually experiencing the shock or experiencing the sound. They've gone further than that, in that they've been able to eliminate a particular memory. So they take mice that have this response, and then they can use CRISPR to eliminate the encoding of that response, and then they don't respond. So there's a lot of really interesting stuff going on with memory, including, and the applications of this research are treating people with PTSD, treating people with other sorts of traumatic memories, in that by manipulating the chemical environment in the brain while that memory is being accessed can change how someone responds to that memory. So giving someone a euphoric sort of drug like propofol while they're recalling some horrible memory doesn't make them feel great when they recall the memory not on propofol, but it makes it much more endurable, much more able to deal with the stress of that memory than without the treatment. Okay. So nitric oxide does other things too. It's a very common signaling molecule. It's involved in smooth muscle contraction and relaxation, and smooth muscle like that lines the lines blood vessels. So acetylcholine will act as a first messenger outside the cell, binds to a acetylcholine receptor, which is G protein coupled receptor, activates its activator phospholipase C. Phospholipase C generates IP3 and DAG, not shown on here. Okay, and you have a release of calcium from the endoplasmic reticulum inside the cell. That calcium is required for nitric oxide synthase to convert arginine plus oxygen into citrulline plus nitric oxide gas. Nitric oxide gas binds to a nitric oxide receptor on muscle cells, generates cyclic GMP, which stimulates protein kinase G, which stimulates relaxation of muscle cells. There, at one time, at the University of Akron, there were people in polymer science working on polymers that would be nitric oxide donors that could be placed on patients that needed constant blood flow to a certain area. Let's say they're someone healing from a burn or something like that. And another aspect of nitric oxide signaling is involved in penile erection. So the autonomic neuron of the pelvic plexus, which innervates the penis, blood vessels in the penis, and the erectile tissue of the penis called the corpora cavernosa. This neuron, when stimulated, releases nitric oxide, which stimulates guanylyl cyclase to make cyclic GMP. The blood vessels relax and they fill with blood, and mammals, anyway, get an erection when that happens. For the erection to go away, you destroy that cyclic GMP. And if you stimulate the nerve and there's no nitric oxide that is allowed to be made because you're using a nitric oxide inhibitor, you get no erection. So you need nitric oxide as a signal for the erection to happen. So the erection goes away when cyclic GMP is destroyed. The second messenger is destroyed. And that thing that destroys the second messenger is called cyclic GMP phosphodiesterase type 5. And type 5 is the type that is expressed in erectile tissue. There are other types found throughout the body, and they have something to do with some people who have adverse reactions to these drugs. Okay. About 10% of human males are impotent, and there's a drug called Viagra. There are competitors called Cialis and Levitra, which work by similar mechanisms. And the way that Viagra works is it inhibits cyclic GMP phosphodiesterase type 5. So the enzyme that destroys the cyclic GMP second messenger is inhibited by Viagra. So all the other parts of the signaling pathway have to be there, and that the male has to be sexually stimulated so that that nerve, the autonomic neuron, is sending action potentials. And cyclic AMP has to be made, but then for people that respond to Viagra, the problem is that the cyclic GMP is made but doesn't stick around. And so this drug allows the cyclic GMP to stick around and allows men who are impotent to maintain an erection. The story goes that this was developed as an angina drug, and I've told that story. And that story comes from that's how the drug was filed for a patent in the clinical trials. And the clinical trials were aimed at treating angina, which is a heart disease problem that is related to restricted blood flow in the heart. And so by opening up blood vessels in the heart, you can treat angina. When they made this drug, they were aware, Pfizer was aware, the company that makes this drug, was aware that nitric oxide signaling was happening throughout the body and through many systems, and that they were aware that it was a possibility that it could treat erectile dysfunction. They didn't know until after the trial that it was reacting specifically with the type of cyclic GMP phosphodiesterase that is expressed in erectile tissue. And that's why they had the dramatic effect during clinical trials that these men who are being tested for treatment for angina had persistent erections. So one of the indications and side effects of Viagra is that you can have a rapid loss of blood pressure. And this is in some individuals who have a different expression of cyclic GMP phosphodiesterase, or their version of cyclic GMP phosphodiesterase reacts to Viagra where in their heart, for example, where most people do not. Or it can be a dose thing. And that some people think, if one little blue pill is good, then 5 is really good. So there are people who are taking this recreationally and have had cardiovascular problems because of this. I also just read a thing that long-term use in young people can lead to destruction of tissue, which is not something that anybody is looking for. But it's a big seller for Pfizer, over $1 billion a year that they make from this drug. Okay. There is also some use of the drug for other applications. So in 2018, there was a study done that a small dose of Viagra daily could reduce risk of colorectal cancer. These are in people have a high risk of colorectal cancer. They have a family history of colorectal cancer. They may have had several polyps removed, and the Viagra was preventing a signal for cell proliferation in early polyps, not an advanced polyp, but in early polyps. And so it's possible that it can be used in lots of different applications. Okay. Another aspect of nitric oxide signaling, which is kind of cool, is in fireflies. Hopefully we're going to see lots of these this summer in our backyards. And this flashing that you see in the firefly is due to nitric oxide signaling. So in insects, they deliver oxygen to their cells in a different way than we do, in that they have a direct connection to the outside air through these tracheals, and they terminate in this structure called a rosette, and the cells are right next to that rosette when the mitochondria are packed up right against that. This is actually a lot more efficient because you don't have to bind oxygen to a binding protein, and you can just deliver oxygen right from the atmosphere to these cells. So the mitochondria are right next to the oxygen delivery system, and which is connected to the outside air. And what nitric oxide signaling does in these guys is there's a little nitric oxide puff that is made by the fireflies in their lantern organs. That nitric oxide temporarily knocks out the mitochondria that are right next to the rosette, in that they can no longer use oxygen as a terminal electron acceptor. They're so-called stunned. That allows oxygen to travel past those mitochondria and be available for nitric oxide synthase, not for nitric oxide synthase, be available for this luciferase reaction, which takes the substrate luciferin with oxygen and creates light as a result of that. So again, what happens is there's nitric oxide synthesized, which also requires oxygen. And then that nitric oxide synthesis knocks out mitochondria. The oxygen can get past those mitochondria and be available for luciferase, which is an enzyme that takes the substrate luciferin and makes light. This reaction is used in biochemical studies, where they use light as a signal. They attach the luciferin enzyme to something that you want to measure, and then you measure the amount of light as an indicator of how much of that thing is expressed, for example, and requires the substrate luciferin, which comes from fireflies. Normally, when they are not flashing, the oxygen doesn't make it much past the mitochondria, is used up by the mitochondria, and there's not enough oxygen available for this reaction, and so they're dark. So when you see a flash like that, that means that each flash is associated with a puff of nitric oxide, temporarily knocking out those mitochondria and making the oxygen available to the luciferase. This is just showing a stain for nitric oxide synthase, and it's associated with these rosettes in the trachea of these fireflies. Okay, so now we're going to walk through a bunch of different pathways, and you're going to, if you're not there already, you're going to get to the point where you're asking, like, why am I learning this pathway? There's so many pathways. Why do I have to learn all these different pathways? The reason is, like I said before, in that it's important to have these pathways in your head so that you understand the vocabulary that is associated with these pathways. At the end of this class, we're going to talk for a few weeks about cancer and how cancer develops. There are many ways that cancer develops. It's not a single disease. It's a phenotype of uncontrolled cell growth, and there's so many different ways that cells can get to uncontrolled cell growth. Many of these pathways are cell proliferation pathways. They're telling a cell, don't divide, or they're telling a cell, do divide. And so knowing the particulars about how it tells a cell do or don't divide will be important for understanding how cancer develops. There are lots of terms associated with these pathways, and eventually you're going to see that they have a lot of things in common. Hopefully you're starting to see that already, like receptor dimerization and so on. But at this point, when I say here's a pathway and here's another pathway, it's going to be hard to understand why this pathway and why not that pathway until we get later on, until we start talking about the cancer stuff and some of the immunology stuff. And as much as I can as I go along, I'm going to try to give you little snippets to try to distinguish between one pathway and another pathway. Okay, so this one is called transforming growth factor family of molecules that are involved in the TGF-beta pathway. So TGF-beta was named for a pathway that was being studied in cancer cells, and it was called the transforming growth factor because it turned these cells from non-cancerous into cancerous, and not only that, into metastatic cancer cells. It was a transforming factor. And like you've seen in other aspects of biochemistry and cell physiology, before they know what it is, they call it a factor. And they didn't even know it was protein at that point. Okay, so in this pathway, R2 is the thing that you've got to get to. So R2 is receptor 2, R3 is receptor 3, and R1 is receptor 1. In some cells, depending on what kind of cell you are, if you're a liver cell or an epithelial cell or a lung cell or whatever, there's a receptor called R3, and transforming growth factor beta, which is the primary signal. This is the thing that's floating around in the blood, the first messenger, is going to bind to R3 first, which will then recruit R2. In some cells, it binds to R2 directly, okay? And in the cells where it binds to R2 directly, then R2 will recruit R1, okay? And so either R3 first, then recruiting R2, and then R1, or R2 directly, then recruiting R1, depending on the cell type, okay? Once you have whatever bound to transforming growth factor, either it's R3 or R2, depending on the cell type, and then recruiting R1, then you have a kinase complex on the inside of the cell. It's going to start phosphorylating stuff, okay? And you've seen that sort of thing before. In this case, the thing that is phosphorylating is called a SMAD. The SMADs got their name from two different names of similar genes found in worms and flies. So it doesn't stand for it's not an acronym. Like TGF is transforming growth factor, SMAD is There's no SMAD. It's two different genes smashed together to make this word SMAD. Okay, so SMADs are different molecular weights in lots of different families and so on. There is a NLS, or nuclear localization factor, attached to the SMAD. SMADs get phosphorylated by this complex after they bind TGF-beta. Once they get phosphorylated, they interact with another SMAD, in this case SMAD4, and they pick up a protein called importin. IMPORTIN. Not important, but importin. Like import, like import-export, and then in at the end of it. Importin is important for getting this complex into the nucleus. So this is like a ticket to get you through the gate of the nuclear pore. The SMAD complex goes into the nucleus, and then there's a protein called RAN with GTP bound to it that takes the importin off once it's in the complex is in the nucleus. And now that the complex is in the nucleus, it's going to bind to DNA. So this SMAD complex is a transcription factor, along with TF3, TFE3, which is another transcription factor, and it will change what genes get expressed. Okay. Again, why am I talking about this? So there's all kinds of human cancers: pancreatic cancer, retinoblastoma, liver cancer, colon cancer, stomach cancer, leukemia, and they lose responsiveness to TGF-beta. They stop responding to this signal, and that this signal has to arrive on cells for the cells to not divide. So if TGF-beta signaling stops, the default will be that cells will go ahead and divide. So when all this stuff happens, okay, recruit your R1 after you bind R2 or R3, and then you phosphorylate your SMADs, and then you get your important, and then you go to the nucleus, and the important gets taken off, and then you recruit TFE3, and then you bind to the DNA. After all that stuff happens, the cell gets the message, do not divide. If this doesn't happen, the cell does not get the message, do not divide, and so the cell goes ahead and divides. So cells that lose their responsiveness to this signaling, they go ahead and divide. Uncontrolled cell division, cancer. Okay? Here's another signaling pathway that you'll see a lot in developmental pathways and also in cancer pathways. And it's called WNT, W-N-T. It's a smash together of wingless and integration, which are two Drosophila mutations, names. And those are the mutants where it was discovered. Okay, so WNT is a first messenger. It's floating around in the blood. It's hydrophobic. It's associated with fatty acids. And that's important for how it moves through the blood. So it tends to be pretty local from where it's produced to where it's acting, and it can form gradients. There's these really beautiful pictures of WNT patterns in Drosophila that are important for how the embryo body plan gets set up. Okay. If there's no WNT, then WNT is not binding to its dual receptors, LRP and Frizzled. Two different kinds of receptors, it binds to both. Okay? In that case, there is a molecule called beta-catenin, which is a transcription factor, and that molecule is multiply phosphorylated on serines and tyrosines by an enzyme called GSK3. And when GSK3 is bound to beta-catenin, with these other proteins with it, APC, axin, and CK1, its activity for phosphorylating beta-catenin goes way, way up, like 20,000 fold goes up when these proteins are there versus when they are not there. When beta-catenin is phosphorylated, it can bind to TRCP, and TRCP is a ubiquitin ligase. Remember, we talked about E3, E1, E2, E3. E3 is the ubiquitin ligase, and TRCP is a type of E3. So this thing gets phosphorylated, E3 binds to it, E3 does its thing, adds a whole ton of ubiquitins to it, becomes polyubiquitinated, and beta-catenin gets degraded, never gets to go to the nucleus. As a result, TCF, which is a transcription factor, and Groucho, which is a transcription factor, TCF normally would activate target genes that it's binding to, but Groucho is preventing that from happening. Groucho is preventing TCF from activating these genes. So no Wnt, genes don't get activated. If there is Wnt, okay, now Wnt binds to its co-receptors, LRP and frizzled, both. When both of those are bound together with Wnt, they attract disheveled. And when disheveled is attracted to this complex, axin is more attracted to this complex. There's a phosphorylation of LRP than it is to normally this beta-catenin complex. Okay? And this whole complex, APC, axin, and CK1, makes GSK lots more efficient. So if axin can't be part of that complex because now it's bound to the Wnt receptor, GSK3 has low activity for phosphorylating beta-catenin. So instead of beta-catenin getting degraded, it builds up. It can now go to the nucleus. It can displace Groucho, okay, and recruit some other transcription factors, PIGO, LGS, and CBD, and now these target genes get turned on. Okay, so with Wnt, attracts disheveled, which attracts axin, because axin is attracted to this receptor. It's no longer activating GSK3, so beta-catenin is no longer being phosphorylated at such a high rate. So the ubiquitin ligase is not attaching ubiquitins at such a high rate. Beta-catenin goes to the target gene, knocks off the repressor, Groucho, and the target gene gets activated. Okay. Again, Wnt signaling is another growth factor sort of signaling. It's activated in, it's important in lots of cancerous tumors. Human tumors have messed up Wnt signaling. It's also important in development. Okay, one more, and then we'll call it quits for this podcast. And this is hedgehog signaling. This is named for Sonic the Hedgehog, the video game, and recent movie with Jim Carrey. This was the video game that the grad student who discovered this thing was playing at the time. And the flies where this mutation was discovered had very bristly sort of hairs on them. It made them look like hedgehogs. And so hence the name Sonic the Hedgehog. The people who study flies have much more interesting mutation names than people who study other sorts of model systems. And usually that gene gets, the name sticks for whatever organism it was first studied in. And that has been a problem for like genetic counselors, for example, who may be telling a family about a mutation that they've discovered in somebody's precious little daughter, and it may be something called like cheap date, for example, is an alcohol metabolism mutation that was discovered in fruit flies. But anyway, Sonic is one of those. There's now a family of hedgehog signaling proteins. There's Sonic hedgehog, there's desert hedgehog, and there's Indian hedgehog, which are all types of real hedgehogs, and also types of hedgehog proteins. The other thing that's cool about hedgehog signaling is it involves this thing called the primary cilium. So in cell molecular biology, you talk about cilia in terms of being at the base of undulating structures for cells, the basis for structures that may beat, the basis for long processes in axons where motor molecules, kinesins and dyneins, can walk up and down these stabilized microtubule structures. But it's also true that all cells have at least one cilium, and it's called the primary cilium. And you don't tend to think about them, and you have to prepare the cell just right in order to see it. But it's important in hedgehog signaling. So hedgehog signaling is a little bit complex, so talk about it here. So if there's no hedgehog signaling, hedgehog binds to its receptor called patched, PTC. And patched, with no signaling, with no hedgehog bound to it, inhibits the fusion of a membrane-bound protein called smoothened from a post-Golgi vesicle ready to fuse the membrane. It prevents that from happening. Because this doesn't happen, there is a kinase that phosphorylates a transcription factor called GLI, and when GLI gets phosphorylated, it gets partially proteolytically cleaved into GLI-R, and then GLI-R goes through the nucleus and binds to DNA, it's a transcription factor, and prevents certain genes from being transcribed. Okay? So, GLI-R, you think R as a repressor, GLI as a repressor molecule binds to these genes and prevents them from being transcribed, and that's patched with no hedgehog bound to it. When hedgehog does bind to patched, now its inhibition of these vesicles with smoothened transmembrane protein in them is no longer inhibited from binding and fusing with the membrane, and so they do that, and they incorporate into the membrane. They incorporate into the membrane of the primary cilium, and they move in the membrane of that primary cilium through the transmembrane fluid membrane, and they inhibit a protein called SUFU. SUFU was important for the kinase activity of these kinases acting on GLI, and so GLI no longer is acted on by that kinase. Instead, it is transported up the primary cilium by a kinesin. So this is a motor molecule that it physically grabs this protein GLI and moves it to the end of the primary cilium, where GLI gets modified and becomes GLI star. It gets modified by phosphorylation, not the same phosphorylation as down here, phosphorylation at different sites, and also other modifications. I'm pretty sure it gets acetylated, but I'm not totally positive by that. But it gets modified, becomes Gli star. Once it is Gli star, it attaches to diene, which is another motor molecule that goes in the opposite direction on these microtubules. Remember, microtubules have polarity, a plus N and a minus N. Kinesin goes in one direction, diene goes in the other direction. Diene brings Gli star down to the base of the primary cilium. Gli star then can enter into the nucleus and activate those genes. So it binds to the same place and affects the transcription of the same genes. But Gli-R, repressor, represses transcription of those genes, and Gli star activates transcription of those genes. So hedgehog binds to patched. Patch no longer inhibits smoothen from fusing with the membrane. Smoothen fusing with the membrane inhibits SUFU. SUFU then is not aiding the kinase activity of Gli on Gli. Gli is then transported up to the top of the primary cilium by kinesin. It becomes modified to become Gli star, transported back down to the base by a diene, and then Gli star enters through the nuclear pore to activate these genes. Okay. The Gli here, becoming Gli-R, Gli repressor, is tagged by a member of the ubiquitin tagging pathway. So again, ubiquitin is involved in this pathway. We actually don't know that for humans yet, but it works that way in flies, and we're assuming it does work that same way in humans. Brain tumors, muscle tumors commonly lack normal hedgehog signaling. So again, it's involved in cancer. Okay, that's enough for one podcast.